So basically, when we talk about heredity, no? So if you go to Mendel, okay, yung mga nasagasaan to sa Earth and Life Science eh. Kahit na hindi kayo nag-STEM, nasagasaan to. It was also tackled, pero very minimal lang yung diniscuss. And when you were in junior high school, di ba, heredity, what are genes, ano pa, and of course, in cell division. But it's very important to distinguish terms. Uulitin ko, science was very exact. There's no such thing as overlapping of terms, okay? You may see terms in a sentence that was used, for example, in heredity, but the words that was inside that concept has another definition, okay? At ganun ang nangyayari, class, di ba? And what is the secret is for you to be able to understand specific concept on specific terms. Now, we knew that each cell of our bodies contains different proteins. So by this time, alam ko, naintindihan nyo na ano ba yung protein. Okay, we have come so many times in that. Okay. Let's know which protein to synthesize out the extremely large number of possible amino acid sequence. So in this question, the possible answer here is enzymes, diba? Okay, there were enzymes, okay, that guides proteins. No? Aside from saying there are proteins, there were also enzymes, and there were even receptors and neurotransmitters. So from the end of the 19th century, biologists suspected that the transmission of hereditary information took place in the nucleus. So you know na in the nucleus, no, where you find the DNA or the deoxyribonucleic acid. And a part of that DNA will be brought up coming from the nucleus to the cytoplasm to process protein in protein synthesis. And that is actually the next lesson. Ito yung huling lesson ng midterm na papasok na sa final term because we lack time. No? So these structures that test or contain DNA are what we call chromosomes. And chromosomes are made up of sister chromatids. Okay, sister chromatids that can be counted depending on what kind of cell division does the chromosomes undergo. Because it can either be divided if it's mitosis or somatic cell division or meiosis, okay, sex cell division. So the hereditary information was thought to resides in genes, no? So yung genes na yun, DNA yun. So, because DNA is just so exact, genes is very generalized. No? But when you say the blueprint of the body, DNA, the blueprint of the body, it's the genes. No? So when you go back to meiosis, we have pairs, no, or split numbers of chromosomes that came from the father and the mother. So now we have this character which is imparted by our parents. No? The fact that it is sexual reproduction, the participation of the egg and the sperm. Hindi naman tayo asexual, hindi naman tayo pinanganak na tulad lang ng bacteria class na nag-divide lang through binary fission. Because that's an example of asexual reproduction. Okay, chemical analysis of the nuclei showed that chromosomes are made up of, okay, mayroon din palang protein ang chromosomes. Yung protein na yon, yun yung histones at yung nucleic acid. So malinaw na sa inyo na lahat halos mula ulo hanggang paa, dinodominate tayo ng isang macromolecule na tinatawag nating protein. Okay. Now, by the 1940s, it becomes clear that DNA carried the hereditary information. It was 1940 going to 1950, di ba? By Watson and Crick. Natatandaan nyo, Watson and Crick. There is someone who also participated here, pero hindi na siya masyadong na-credit kasi ay, nabigyan ng credit kasi ay namatay na siya. Namatay siya dahil sa sobra ding exposure niya sa X-ray, sa kaka-check ng DNA. Ganun naman klase ang invention eh. But now, it's very, ang dami ng scientist. Parang ang hirap na ngayong maging sikat sa isang klase or field of science. But if you wanted to, your course can also lead you to that, you apply in the OST, Department of Science and Technology. If you had a heart of being a researcher, i-pursue nyo yan. Kasi sa totoo lang, iba talaga ang learning ng pagiging researcher. Or either way, you practice nursing first and you check on your passion. And you maybe wanted to get in. So if I will be given the chance, I should have done that earlier. Kasi ang pagiging researcher ay kailangan talaga you have to start young. Okay, para makadevelop ka ng name mo. Okay? Thus, the expression of genes in terms of an enzyme protein would lead to the study of protein synthesis and its control. Now, in nucleic acid, tulad nga nung sinabi ng video kanina, dalawang klase. Kaya pag sinabing nucleic acid, hindi lang yan DNA class. You have to consider RNA. So it's two kinds of nucleic acids, are DNA and RNA. Okay. Pareho silang polymers. So alam natin polymers, it's a complex macromolecule or it's a macromolecule coming from a monomer. A monomer, balikan natin. Ano ibig sabihin ng monomer? Basic unit. Halimbawa, amino acid for protein and monosaccharide for polysaccharide. Okay? So these are nucleotides. Ang nucleotide naman ay binubuo ng base, no? Kanina pinakita ha, monosaccharide and a phosphate. So when you talk about monosaccharide, we are pertaining to the ribose. Okay, ribose sugar. Okay, pakipatay na lang yung ilaw. Okay. So we have here the bases. We have two actually, pyrimidine and purine. Medyo mahirap to tandaan, no? Okay. So when you talk about pyrimidine, okay, it's made up of, pinapatay ko class yung ilaw, ha? O yun naiinitan. Okay. Pyrimidine are made up of cytosine or C, big C to. It's not small c. Thymine and uracil. So you can found cytosine and thymine cytosine both in DNA and RNA only. While thymine, DNA, uracil in RNA. So thymine and uracil is a distinguishing base that you can see and you can identify which is DNA and which is RNA. And of course, we have the purines or the purine, no? We have adenine, and guanine, which is both found in DNA and RNA. So yung distinguishing base lang pala ay nasa pyrimidine lang. Okay. So again, ulitin natin, thymine, DNA, and uracil in RNA. So what were nucleosides? So when you talk about nucleosides, it is a compound that consists of D-ribose. O malinaw na sa inyo, bakit D ha? O, so babalikan nyo yung carbohydrates kasi baka tanungin nyo na naman, ano na yung D-ribose? Okay, you go back. Class, pag sa science, lalo na sa mga major nyo, you always go back. Yung babalikan sa Google. Kasi nag-overlap naman talaga ang information, di ba? Nakakalimutan nyo talaga yan. At hindi ganun kadaling magkaroon ng retention. I would say if you're photographic, you have the photographic memory sa psychology, swerte mo. Pero alam nyo ba, pag photographic ka, dumadating din yung time na nakakalimutan nila. I was not that sure if photographic memory is processed in the short-term memory. Ako kasi class long-term ako eh. When I was younger, I had that photographic. When I grow older, nag-shift ako sa long term. Ano ibig sabihin nun? Hindi ako ganun kabilis mag-grasp. And I would say there were really students who are like that. But you will just be amazed in your entire life na biglang naalala mo yung sinabi ng prof mo, ang tagal na, pero nung exam hindi mo siya naalala. So you see, wala namang mali talaga. Nagkataon lang na late lang talaga na ilagay ng mind mo sa long term. The advantage in long term, for example, in my case, in teaching. Because if all information was placed in the long term, wala ka ng problema. Pag nagturo ka, because you just have to recall one word from there and makikita mo na mapoprocess mo na yung information immediately. Okay. But of course, when you talk about photographic memory, this is very exact naman, no? Kaya lang book talaga lang class. So when you're a teacher and you have a photographic memory, you should know how to localize information. When you say localization, this means you have to give or understand or try to comprehend basic examples to relate to students. Okay? So this D-ribose or 2-deoxy-D-ribose, to be exact, is bonded to purine or pyrimidine. What bond? Okay, beta-N, glycosidic bond. Glycosidic bond, to recall, is the bond that was used to bond carbohydrates. So here, wala naman tayong pag-uusapan class, but you have just to look at the structure. Okay, this is an anomeric carbon. That's why it's a beta. It was linked to the beta-N glycosidic bond. Okay, and here is a beta-D-riboside, no? This one. Okay, and this is the uracil. So sa DNA, mahalaga na alam mo kung ano itsura ng uracil, a ribose, okay, the position of the glycosidic bond, and of course to recall on what is an anomeric carbon. Now, when you talk about nucleotides, is a nucleoside. Okay, so you see, a nucleoside is different from a nucleotide. Baka akala nyo, wrong spelling lang, ha? Okay, so a nucleoside in which a molecule of phosphoric acid is esterified by a hydroxide of the monosaccharide. Okay, either on the 3' or the 5' hydroxide. So kanina nakita nyo, no? Boseman explains that there's a 5' and the 3'. 5' and the 3' is actually the beginning and the end of the double helix DNA, no? Double helix DNA. And it was positioned anti-parallel. Kung ito yung 5', 3', ito yung 3', 5'. Anong dahilan? Bakit ganon? Yun ang reason, class, kaya hindi sila nagkakaroon ng tangle o nagbubuhol-buhol. And even if, we'll check if I could show you DNA replication later. Because I think that was also important. No? Na yung lagging, the lag strand, i-recall ko, the lag strand, okay, has its opposite position. Kumbaga yung duplication niya, class, pabaliktad. So in that case, di ba, sa cell division, hindi talaga nagkakaroon ng pagbubuhol. Kasi kung isipin mo, kung pantay sila. Okay? So maraming reason for that 5' and 3' and 3' to 5'. But you will just be amazed, no? Because man was created before science. Di ba, class? You go to the Bible. No? You don't put science. Kung wala nating iisipin yung science, no? It's a very amazing, we are actually a very amazing invention or a discovery, okay, for the human species. Kasi hindi lahat, okay, para sa information, hindi lahat ay mayroong ganun katulad sa atin. Okay, how our DNA replicates, no? So kung curious kayo, you check on, since we have the same cells with mammals, no, the highest form sa kingdom ng mga species, ay i-check nyo kung tayo lang ba. Kasi hindi tayo masyadong subjected sa mutation. The fact that we have DNA. Kaya nga, di ba, yung virus, the virus is acellular, no? It's not considered to be cell. Kaya ang bilis niyang ma-mutate, mag-change from one kind of RNA to another because unang-una, wala na naman nagpuprotect sa kanya eh. Siya ay isa lang na genetic material. Sa siyang RNA. Na pag na-subject siya sa environmental changes, nagpunta siya ng isang host, another host, then obviously, di ba, mababago siya. Pero tayo talaga hindi dapat. Although we knew that we have cancer cells, but the mutations that our body should undergo should be very slow because kaya nga semi-conservative ang tawag sa DNA replication kasi ay hindi siya madaling magkamali, no? So, this is the deoxythymidine 3' monophosphate 3D-TMP. Okay. So nucleotides, so we have adenosine 5' triphosphate. Of course, you are now familiar with ATP. Serve as a common currency into which energy gained from food. So if you're being to ask, does the DNA has the ATP? Yes, no? Okay, but it's very exact. Adenosine 5' triphosphate. Okay. So how it was produced, okay, AMP, ADP, and eventually it becomes an adenosine 5-triphosphate or ATP. So here, the oxygen here are anhydride, and the oxygen here is an ester. Or you talk about ester, di ba? You go back to the organic compounds or different functional group in the organic compounds. So let's go now to the structure of DNA. So you see here an schematic diagram of a nucleic acid molecule. The 4 bases of each nucleic acid are arranged in various specific sequences. So ito na yung sinabi ko kanina. Okay. So kung single bond lang, no, single muna, hihiwain natin sa gitna, no? So this needs to be paired. Okay? G, nakikita nyo na para siya maging pair, ito yung shape na kailangan niya. Diba? And then for adenine to be pair, it should be with thymine. So it's very exact to fit in the shape to have the base pairs. So pwede nating tanungin, ma'am, paano yung sequence? That's very unique and that's only very specific for an individual. No two individual would have an exact DNA, even for twins class. Of course, for cloning, that would be different because in clone, exacto. Because it's your clone there. It's an exact replica of your DNA. Pero for twins, even for siblings, walang exacto class. So, DNA secondary structure, the ordered arrangement of nucleic acid strand. The double helix model of DNA secondary structure was, of course, remember this, Watson and Francis Crick, no? James Watson and Francis Crick in 1953. Dito nagsimula ang revolution ng pagkakadiscover ng exact, what we call the structures of the cell, what really dominates us. But if you're concerned, buntik na itong ma-oppose class of that term, prions. No? Prions is an infectious protein. Okay, just have to check that out. Prions, P-R-I-O-N-S. No? Buti na lang, nag-split pa rin sila sa gitna. Kasi kung nagkaroon ng prions, yung Kuru disease, I don't know if it's in 1990, na tayo pala bilang tao ay pwedeng ma-infect ng isang klase rin ng protein that is also a part of us. So that prions actually emerged from the Kuru disease ng cannibalism yan eh. When a tribe ate the brain, no, of the, I don't know if the head of the tribe o yung pinakamatalinong, but this is already dead, no? So, I think this start with a disease, yun nga yung Kuro. Kaya nung tiningnan nila to, teka, paano mangyayari to? And the fact that we are also made up of protein, the DNA, lahat yung buong pagkatao natin. Kaya nga sabi ko, hindi dapat mag-dominate ang prions na to class kasi tatalunin nito lahat. Because how can you kill that when it's also a cell that is part of you? Okay. When you consider cancer cells, you inhibit cancer cells, then you inhibit also the good cells, di ba? For chemotherapy. But when you inhibit prions, meaning, or proteins that is inside your body, meaning you are going to inhibit the entire protein of your system, then that's more dangerous and it would really risk life. Buti na lang, hindi na ito lumaganap at sana hindi na class. So, ano na, saan ito nakuha? Nung kakain sila ng cadaver. It's actually a part of the brain. Okay, so it stops. No? You wanted to do research about this class. Basahin niyo yan. Kuro disease, prions, or I don't know, puro lang ang nag-evolve, no? Baka may iba pa. Okay? So, the DNA double helix, we have this. Okay. So, pag DNA, meron siyang dalawang klase ng groove, the minor groove and the major groove. Okay, so if you're going to look at this is the minor and this is the major groove. Okay. So, in base pairing, okay, sorry. Sa base pairing naman, mahalaga na makita nyo kung nag-move masyado ang aking PDF. Okay. So this is how, so there are hydrogen bonds. Which connects them actually are hydrogen bonds. No? So this is a thymine-adenine pair. Dalawa lang siya, no? But when you talk about the guanine and cytosine pair, there were 3 bonds that connects them. So DNA is called around protein called histones. Histones is also one major reason why the replication is very efficient, very effective. Parang accurate sobra. Kumbaga sa sinulid, hindi yung buong sinulid, class. May bilog-bilog na nag-hold ng bawat DNA structure. So meaning, from one histone, magsusukat na naman another histone and another histone. So nakita nyo, bakit hindi siya sobrang nagbubuhol ng buo? Okay. So these histones are rich in the basic amino acid lysine and arginine, whose sides have a positive charge. Tandaan nyo ha, the sides of the lysine and arginine, which hold histones, okay, are positive charge and they are basic amino acids. So the negatively charged DNA molecule and positively charged histones attract each other. So if there's a negative charge, therefore, what would attract it? The positively charged, no? And form units called nucleosome. What is a nucleosome? A nucleosome is a core, bilang, 8 histone molecule. Okay, bilangan. So one nucleosome, another nucleosome, another nucleosome. So tandaan nyo, in one nucleosome, there are 8 histone molecules where the DNA helix is wrapped. Then, this nucleosome will be further condensed. Chromatin. Okay? And these chromatin fibers, when organized into loops, and the loops into the bands that provide the superstructure we call now chromosomes. Okay? Malinaw ba sa inyo? So, from histones to nucleosome to chromatin to chromosomes. Pero kasi, lagi na lang sinasabi, okay, sister chromatids, okay, chromosomes, but in biochemistry, it's precise. So, measurement-wise, yung width lang, no? The width, 2 nanometer, but the nucleosome, pag kinoil, ang isang nucleosome is 11 nanometer. Okay, that's the width class. It's not the length. No? Okay. So, chromosomes are beads on a string. Okay, 11 nanometer. Pag ito naman ay 30 nanometer. So, there's another term. Solenoid is made up of 6 nucleosome per turn. Tingnan nga natin. 6 nucleosome per turn. Okay, may exact numbers ba dito? Wala, no? Okay. So, solenoid. Next are, okay, when you talk about now chromosomes, it's a loop of 50 turns per loop. Okay. Of course, they have the base, the matrix. This is now 250 nanometer. So kung titingnan nyo, from a very small measurement, it becomes bigger or larger in terms because of naging chromosomes na nga siya class, no? Okay. So now we have a mini bond of chromosomes that is in 18 loops. Okay, this is now an 840 nanometer. And the stacks of these mini bonds now will give you a chromosome. Okay? So, isipin nyo kung gaano kahaba, what was the length. Sabi nga nila, if you're going to stretch all the chromosomes that you have inside your body, eh, milya-milya ang aabutin. So, what is the difference between a DNA and an RNA? So, the three differences in structure between DNA and RNA are, okay, DNA bases are adenine, guanine, cytosine, and thymine. The RNA bases are adenine, guanine, cytosine, and uracil. So, the sugar, okay, in DNA, we have 2-deoxyderibose. Walang oxygen, yung number 2 carbon ng ribose na meron sa DNA. And it's present in the ribose of RNA. DNA is double-stranded and RNA is single-stranded. So I think I'll end there. We still have 19 slides to go. Yeah.